Oh, you're in mute.
I'm muted. Sorry. Yeah, sorry, I was muted. I didn't realize. Welcome everyone to today's seminar. I just want to make sure that you can see my screen if someone can confirm quickly the presentation.
Yes,
Yeah,
we can.
perfect. Perfect. Welcome everyone. Today's seminar is on the topic how modern research teams work today. And first of all, I would like to thank the Institute of Chemistry, Metallurgy, and Technology in Belgrade and Professor Olga Jakšić for enabling us to organize this seminar for you. My name is Katarina. I'm from CDD Vault, and I'm here with my colleague Susana Tomasio, who is one of our application scientists. And she will actually show you how the platform is used in practice by scientists like yourselves. Before we jump into Susana's presentation, I just want to say a couple of words about CDD Vault as a very quick introduction. CDD Vault is a cloud-based solution for managing chemical and biological data. So that means that you can digitally register your small molecules, but also biological entities, as well as formulations, for example, in cosmetics and even structuralist entities. So it's applicable to different areas. It's widely used in medicinal chemistry. However, like I said, applicable to different areas, as you can see here on the screen, and also used by biologists, not only chemists. As you can see here on the next slide, CDD Vault consists of multiple modules. That means that the platform is modular and you can pick and choose which modules you would like to use in your research, which ones are applicable to your research actually and useful. Susana will talk more about these, so I won't take more time explaining them. But what I would like to mention is that everything revolves around registration and activity modules. That means that once you have your entities registered in the system, for example, small molecules, along with your experimental data, you can use other modules as well. And lastly, I would like to mention two academic examples. CDD Vault is used by more than 700 organizations to date across the industry and academia. But here you can see two examples of two institutes that are using CDD Vault, Institut Pasteur and IOCB in Prague. You may be familiar with these institutes. So for example, Institut Pasteur uses CDD Vault as their central repository to manage their compound libraries and screening data, which allows them, given that all of their data is centralized in one location, so they can manage it and analyze it, they can compare compounds and assay results, which then allows them to identify anomalies early on rather than later in the process when it's a lot more difficult, which improves their data quality but also research outcomes. IOCB, for example, they used to use Excel to manage their data, which meant that their workflows were painful. That means they were manual, took a lot of time, they were prone to errors as well. But because they implemented modern tools, what took them days in managing their data now takes hours. So it saves them a lot of time. But also modern tools ensure the data is fair, which is one of the more important principles in science. And it's becoming even more relevant in science today. That means the data is easily findable, easily accessible, interoperable, and reusable. And I won't take a lot more time because Susana needs to show you actually the system and how it's used, like I said, in practice. In the meantime, I will share in the chat, I will share a couple of links. I will share links to the full case studies for IOCB and Pasteur if you would like to read more on those. I will also share a link to the free visualization tool in CDD Vault that you can use after the seminar and test out yourself, see how scientists use it to analyze data and present data. If you cannot access it for any reason, please do let me know. My email address will also be in the chat if you will have any further questions after the seminar. Also, once Susana is done with her presentation, we will switch to the Q&A session. So during the seminar, you can ask any questions, type any questions in the chat box, and Susana will answer your questions. Or at the end of her presentation, you can unmute yourself and just ask her directly, whatever you prefer, whatever is easiest. So I will now stop sharing because Susana needs to share her screen. I will hand it over to her.
Thank you, Katarina. Hi, everyone. Thank you. So let me start sharing my screen. So my name is Susana Tomasio. I'm a senior application scientist at Collaborative Drug Discovery. And so I'm basically part of the technical team and I provide technical support to the CDD Vault users. So I will be doing a general kind of a overview of CDD Vault, and then at the end we'll have some time for questions. Right, so what I'm showing here at the moment is our website. So CDD Vault is an online platform. There is no need to install any software in your computer. So all you need is internet connection and a modern browser. As Katarina mentioned, so CDD Vault is used for managing drug discovery data. Well, data, so research data. It can be used for the registration of entities. That can be your compounds, your proteins, your antibody drug conjugates, and then all of the associated data. So when we log in using the email address and password, we reach this landing page, which basically shows the name of the vault or database and then the projects. So central to CDD Vault is the concept of projects. We can have multiple projects in the same vault, in the same database. The users will only be able to see the data and the compounds or the entities of the projects that they are part of. I will come back into this page in a moment. First, I would like by starting with our visualization tool. I would like to mention that our visualization tool is free for all users. Katarina will add the URL for the visualization, which you can access. And basically, you can upload an file or an Excel sheet or an SD file with your compounds and data, and you can do this type of plots that we see here. So basically, this is the URL that we will share in the chat. And then when you upload either an SD file or XLSX or CSV, you are able to view your data, filter your data based on different filters, as we see here on the right-hand side, and just add different types of plots. So what we are looking at here is a 2D scatter plot where we can see the structure for each data point. In the x-axis, we can change the axis basically to any of the parameters as it was brought from our Excel sheet. And then similarly in the y-axis, we can also adjust the axis as well. In the settings tool in here, we can add 3 extra parameters into color, shape, and size. So here we have the log P, the log S, and molecular weights. And we have all of these different plots here available. So let me just zoom in. So now we can see the bar charts, the histogram, and also the actual radar plot. There is a double arrow here at the bottom where we can see the actual table of results with the data that was uploaded from the file. We can see the structure of the molecules, and then we can also color code the table using the settings at the top here. And basically, we can color code based on what the ranges are interesting. So in this case, red means that it's not active into this target. Green means that it's active. So this just allows us to look at a list of compounds and see quickly which compounds are active or inactive into a particular target. But of course, this can be used to any types of data, really. Okay, we'll come back into the visualization. Again, I just wanted to show you that this and mention that this is free. You can use it today by accessing this URL. Okay, so now when we are actually inside CDD Vault, We can have, as mentioned, we can have multiple projects, and we can register small molecules, the structure of small molecules or other entities in those projects. We can also register the assay data or the data that we get from our experiments, and then we can perform this type of query searches where we are searching in 3 different assay protocols. We are looking at all the compounds with the furan search substructure, and this is the results of the query search. The advantage of having the data structured and registered in this system, in CDD Vault, is that we can query the data. This report table shows on the left-hand side the structure of the compounds, and highlighted is the substructure that was used in the query search. And then we can see also the properties, basically the chemical properties, around 20 chemical properties are calculated whenever the chemical structure is registered in the system, and we can see the molecule and batch information. In essence, when we register a compound, it will be assigned batch number 1, but if the compound is synthesized again, or if we purchase another batch from a different vendor, for example, this would be batch number 2, and so on. So that information is captured in what we call a batch field, so batch-specific information. Then moving towards the right-hand side, we can see the assay data associated with these compound batches. The first assay is some percent inhibition to different cell lines, and what we are looking at is the average over the 3 replicates that were tested for each batch. We can also see a link to an ELN entry. CDD Vault includes an electronic lab notebook, and if the data is registered from the ELN, CDD will automatically generate the link from where the data came from. And we can click on that link. It's already opened in here, and we are now in the ELN, Electronic Lab Notebook. We can see the title of the experiment, the information, so all of these fields are customizable in your Vault. And then this is the actual canvas where we have the materials used, the structure, and then the actual procedures, so how the assay was performed. So this is all free text. We can use all of these tools to add all of this information. You can attach images. We even have an annotator tool which allows us to highlight specific areas in the images or add text, and that will be available in the canvas as well. Now, what's important here is that we can attach an Excel sheet with the actual data, and if the compounds are already registered in CDD Vault, and if we have already an assay protocol created, we can import the file, register this data in the structure side of the Vault, and the data will be automatically linked, as we can see in this table. So coming back into the ELN entry, this is just to show you that this data was imported from the electronic lab notebook, and that's why we have the link to the ELN from the registration side of the Vault. The ELN, or the electronic lab notebook, is basically used in the same way as you would use your paper notebook, but in this case we have the ability to register the data in the structure side of the Vault and have that link back to the structured data in CDD Vault. We'll come back into the ELN in a moment. First, let's just continue here on the right-hand side on this table. Next, I'd like to mention that we can also register the raw data from our experiments. And CDD Vault can generate these plots. In the case of dose response data, if the concentration and raw data is uploaded into CDD Vault, CDD can generate these curves. These curves, these plots, they are live images. I can click flag outliers and override. This opens what we call the plot mini app, and this is where we can QC the data. These crosses here, these are some points that I had already flagged as outliers, but I can re-add them, and maybe I want to flag these 2 points over here. When I click save, the curve is now regenerated, and the IC50 and the plot are automatically updated in the report table. The IC50 value here, you see that it's underlined and italicized. And if we hover over the value, it says that the value was calculated excluding outliers, just to show us that, yes, this data has already been QC'd. Now, important to mention as well that you can upload images or PDFs from your experiments, and then we can view them in this tabular view. And of course, we can use CDD Vault for the upload of single point data. If you have already some endpoint values, those can be registered, and it's in what we call a protocol. When we have a protocol like this, we can click in Customize your report, and here we can see all of the fields available, all of the properties. So maybe I wanted to see the log S as well, all of the molecule fields and batch fields. And basically, yes, when I click update the report, these extra fields are added into the table as a new column. Okay, let's now take a look at a compound that is registered in the system. So we can take a look at the registration. CDD Vault can be used for the registration of your entities. So this can either be the small molecules if you are working with small molecules, but it can also be cell lines, proteins, RNA, antibodies, and it will create automatically an ID. In this case, if the chemical structure is new, a new ID will be automatically generated. And also we will populate the molecule fields accordingly with the data, with information from our molecules. We see here the properties that are automatically calculated when the structure is registered in the system, and we can click on this question mark and we can find more information about these properties and how they are calculated. Okay. Batches tab, this will show how many batches we have for each compound. When the compound is first registered, it will be assigned batch number 1. You can specify any batch information. Even the analytical data can be registered in here. So if you have your HPLC or CLMS, LCMS data, that can be registered also here at the batch level. But if the compound is synthesized again, or if it's purchased again from another vendor, then this will be batch number 2 and so on. Salts are captured in this salt field. When we register the compound, if the salt is with the main structure, CDD will automatically strip the salt out of the main structure and capture the salt in that field. Now, we see here this samples tab. This is for the inventory. Where do I have my batch? How much do I have left? The sample level inventory allows the users to keep track of the location and the current amounts, as well as the expiration date, or even the barcodes or the sample type. All of these fields, they are customizable. So this allows you to keep track of the history of your samples. Maybe you change the location, maybe you make a debit, or maybe you send that sample elsewhere. So that can all be captured in this samples tab. And then in the links, this will show us all the links to the electronic lab notebook. So this is showing us that this compound here has been referenced in these 3 ELN entries. Very important because we always have the traceability of our entity in CDD Vault. Also, how many assays have been run on this particular entity. Currently, 4 protocols or assay protocols have been run on this particular compound. And I can click through a particular protocol and we can see the date of the experiment, which batch was tested, and the actual readout. Now, I mentioned projects right at the beginning of the session. This is very important and it's one of the key, it's central to CDD Vault because you can have your collaborators in the system. They will only be able to see the compounds and data of the project that they are part of. For example, this compound here, we can see which projects have access to the batch. Batch number 1, the members of CRO1 and drug discovery projects can see the information of batch number 1. But for example, batches 3, 4, and 5, only the members of the drug discovery project have access to those batches. The members of the CRO projects not even know that those batches exist. Okay, so the first step in the workflow is the registration of the molecules or entities, and that can be done manually by creating a new molecule. You can launch the structure editor. This is Sketcher. You can draw the molecule using these tools, or even copy and paste from another editor. You can paste the mol or the SD file in here, or even retrieve the structure from a cast number or a common name or an identifier from the vault. So that will just retrieve the structure. Once we have the structure, we will add the molecule information, the batch information, and then create the molecule. Of course, this is useful for the scenario of registering one compound, but for bulk registration, if we have a chemical library, we will use the Import Data tab, where we can register these compounds or entities in bulk at the same time. And that is kind of the first step in the workflow, is to register our molecules or entities. We see here that we have not only molecules, but also sequences, because we can register different types of entities in CDD Vault. The next step is the association of assay results, registering the assay results associated to the compound batches. I chose this example here, the cell proliferation dose response. All of the assay data is associated to the date of the experiment. So this is the run date, which can be the date of the experiment. Now, in the protocol details, we can see the definition of the protocol, the details, and then the actual readouts. So each readout is a container which will read the data from our Excel sheet. In this particular example, it will read the concentration, it will read the raw data and perform a normalization against positive and negative controls, and then it will perform an IC50 plot using the Hill equation, where we plot the concentration versus the percent inhibition, and then it calculates an IC50. There are other readouts in here where in this case, it's a calculation. It just performs the PIC50 calculation, which sometimes can be more useful. And also it stores an image You can store images from your experiments or PDFs by using this data type file, and then we can upload all the images in a zip file. If you're uploading plate data and we have controls, then we can define where the controls are in the plate using this edit these layouts so that the normalization will be performed accordingly. Now, this example is, of course, an example more for the drug discovery, but you can use this concept of a protocol to upload any type of data that you get from your experiments. So if you have your if you have endpoint values, we can create a protocol where we will have, in this case, we have some solubility values here where we can just create the readouts, and then this will read exactly what we have in the Excel sheet. Once these readouts are created, the data can be uploaded into CDD Vault. We will map the fields to the readouts, and then the data can be queried. So to perform a query search, so once the data is registered in CDD Vault, we use this Explore Data tab and then search, and we can search in any protocol that is available in the system. Maybe I want to search in my cell proliferation dose response. And you have the option to search in any run or a specific run, or even the runs added in the past 7, 30, 60, or 90 days. But you can also add other protocols. You can search for compounds which have been tested in this protocol and maybe also in the inhibition protocol. And we have these Boolean terms to choose from. So maybe I want to see all the compounds that have been tested in one of these assay protocols. We have this launch structure editor, which allows us to perform a substructure search. So here, if I launch the structure editor, and if I draw a substructure, so maybe furan, use this structure. Now I click search, and this is performing a search on the fly in CDD Vault, and it just gives the report, so 10 results. Highlighted is the substructure that I used with data in one of the protocols. And of course, we can customize the report to choose which fields we want to see in the table. Once that is selected, the table is now will have the columns that I selected, they are now enabled in the report table. Very important is that you can always export your data either into Excel or CSV or SDF file. You can also create a collection. So this is a collection of 10 molecules which can be shared with the members of your project. So if you are working on a project and we are performing this type of query search in CDD Vault, we can, instead of sending an Excel sheet to our colleagues via email, we can create a collection of compounds, and then there is a collections tab. Let me just open that in here, where we can see all the collections or just the collections I created, or maybe my favorite collections. I can then click share and I can send that. This is a link to the collections tab, and I can either notify everyone or maybe I'll just send a couple of users the notification to take a look at this collection. But this is a secure way of sharing a collection of compounds with the members of the project. The other thing is that we can save a search. Saving a search will save the query search and the report layout, and it can be used anytime. So, and also if more data has been added into the system, these will retrieve more results. And saved searches are available from the Saved Searches tab. Again, you can see all the saved searches or just the ones you created, or if you have any favorite searches, you can flag them and then click search each time you want to see if there is more data available. Okay, so this is the first part that I wanted to show you. So that's kind of that is the registration system. So and the visualization, and so far we have been. I've only showed small molecules. I will show you in a moment that you can also use CDD Vault for the registration of other types of entities. But what I'd like to show you now is our AI module. So still if I just go here into the molecules and I'm just going to open a new molecule here. So the workflow can be, I have a compound of interest and I want to find similar compounds in chemical public collections. I can do that using a fast similarity search, which uses deep learning. These two links here are part of the AI module. We have the find Campbell patented and commercial molecules using deep learning similarity. When I click this button, this is now performing a fast similarity search in around 11 million compounds. The advantage is that this is a secure search. Your compound is not exposed outside of CDD Vault because we have an instance of all of these collections. These are the similar compounds. In the filter by collection, you can see all of the collections available. Currently, I have the enamine selected, but I can select all of the other ones, and now we can see more results. The links, you see here that all of these similar compounds, they are available in Campbell. You can click the link and this will take you into the Campbell website outside of CDD Vault, but the search is secure inside your vault. You see the similarity score, you see some of the properties, and if any of these compounds is interesting, you can actually register it in your vault. And this will take us into the Create a New Molecule page where we can register the molecule. So let's just go back. Also, in the similarity settings, here we can choose the number of hits per collection, which properties you want to see. So I just added a few others. So now we can see other properties in here. You can download the search results. These will download a CSV file with the smiles and the properties and similarity score. We can group by scaffold, and also we can take all of these results into the visualization tool. So the visualization tool that I showed you at the beginning of the session is now also available for the compounds that are found using deep learning similarity. And this is where I can maybe search or filter the compounds by similarity. I can maybe kind of pin a molecule here, and we can add other plots as well. So this is coming from the AI deep learning similarity search. The other option that we have available in our AI module is the ability to get bioisosteric suggestions using this deep learning similarity approach. Clicking this button, this will show us our reference molecule, and now I have the option to select a fragment to replace. These fragments are done based on the BRICS rules. So now I selected the fragments, and we can then look at the results that are seen, well, at the bioisosteric suggestions. This fragment was not a good option. So let me just kind of find another, and then we can click expand, and then this will retrieve other bioisosteric suggestions. Again, we have the option to download the results, to increase the number of hits per fragment, and also select which properties we want to see. And of course, all of the results can be taken into the visualization as well. Okay, so this is for the deep learning AI module, where you can perform either a fast similarity search or get bioisosteric suggestions. We've looked at the sample level inventory, and I wanted to show you here the inventory tab, which is this one here, where we can see all of the samples available in our inventory. We can see, actually we have these edit columns, which allows us to choose what we want to see in the index page. But in this case, we can see the current amount, the location as well for each sample. And that is part of our sample level inventory, where you can keep track of all the barcodes, locations, current amounts, expiration dates, and so on. So next, I would like to further show our electronic lab notebook. So I briefly showed the electronic lab notebook earlier and showed the link between the registration system and the ELN, meaning that if the data is registered from the ELN, the link will be automatically created. Now, when we go into this ELN tab, we can see all of the ELN experiments available. So just as the pages of your notebook, you can see your ELN entries. There is an ID, which is automatically generated, and here I can see all of the experiments. I can search by keywords. So this is great because this will search this keyword in all the ELN entries, and that will search in the title, in the body of the text, even in the files attached, you will get the search as well. We can search also by substructure. Clicking that benzene shape icon, I can add a substructure, and this will search all the ELN entries or experiments that have that particular substructure, and then it will just show us in this index page. Okay, so there we go. And of course, I can edit if I want to show the structure, I can also edit this index page. Moving, I'd like to show you basically the ELN can be used both for chemists and biologists. In this example here, we have the ELN used to capture a chemical reaction. This is actually the final step of a chemical reaction. And this is where we have the chemical reaction in here. I can click through and we can see the chemical reaction in Catcher. We can make any edits. And we see also the stoichiometry table. The stoichiometry table is fully customizable. We can change the mass, we can define which reagent is the limiting one. And also you see here that this reagent or this component here, it's this particular sample, and there is 1 milligram left in that particular sample. The actual product in here can be registered in CDD Vault directly from the electronic lab notebook. So clicking register a new molecule takes us into actually I will show you, it takes us into the create a new molecule page, and then we can register that molecule in the CDD Vault, and we'll get that link directly into the ELN. We have the procedure. We can attach some images, and of course we can attach a file, import the data directly from the electronic lab notebook as we saw earlier. At the end of the experiment, we may want to add a timestamp. So this is that this just captures the local time and the date, and then we may want to finalize. So in this case, I can click finalize and no further modifications will be allowed, or I can enable the witnessing workflow, and when we finalize, we assign a witness so that it is sent into another user to review the experiment. Once it's finalized, no further modifications will be allowed, unless if you need to reopen the experiment, then the Vault administrator, which is a super user in your account, will be able to open the experiment. We have the table of contents in here. We are able to add comments using this button in here and alert users. So maybe I can add another comment. With the add button, I can alert a user, and maybe we are just asking them to please check a particular section. And then when we click send, the user will receive an email with a hyperlink to this experiment. Also, we have the audit trail. Very important. Here we can compare different versions of the ELN. And so I have the current version in here. I have an older version in here. We can compare it, and it also allows us to revert or restore a previous version. Okay, so this is an example of an electronic lab notebook for chemists, but of course the ELN can be used by biologists as well. So we have here an example of an ELISA assay where we see the purpose. We have some materials here, and all of these materials, they are links to entities that I have registered in my CDD Vault. And then I can follow the link and take a look at the details of those entities. This is a file that I attached, and then we have the actual details of the experiment, the plate map, and of course, once we attach the Excel sheets, again, the data can be imported into the system in the structure side of the Vault so that it can then be queried. We can attach plots, as we see here. These are just images. And also we can, yes, in this case, we can also attach any mol files or PDBs, and you'll be able to visualize these in this view. Okay, so that is for the electronic lab notebook. So that's this tab in here. And what I'd like to show you now is that CDD Vault can be used also for the registration of other types of entities. What we see here in this table are different entities registered in CDD Vault and also different types of curve fits available. Depending on your data, depending on what types of assays you're running in CDD Vault and what types of entities you are working with, instead of a small molecule, as we see here, you can have, for example, an antibody drug conjugate or an antibody. The idea is the same. It will register a unique ID, and then we can have all of the data associated with those as well. And in this case, we have different types of curve fits. Right. So we've done kind of a tour of CDD Vault. I just would like to show the visualization one more time because I feel that's quite important, and also you could, since you can use it free of charge, it might be useful. In this case, I have a query search here, I have different compounds, over 166, and different properties and assay data. I have some percent inhibition to different mutants. I can then launch the visualization, take all of these data points into the visualization. By default, this opens a new tab with a 2D scatter plot. The data table that I showed you is down below in here. And then we have different filters which we can use to filter the data. In this drop down, I can select another filter and then use that to filter the results. I can add up to 4 plots in one page. So we can currently only add up to 4, but you'll see that all of the data points, all of the plots are in sync. So if I select a small portion, all of the other plots are automatically updated. The settings in here allows to add other parameters. So the color to the molecular weight, the shape to the log D, for example, and then the size to the log S, for example. And then also we can maximize this table here. We can see all of the list of compounds. We can use these settings to color code, maybe by molecular weight, and then I can just define the ranges that I want. So maybe just like that. So then we can just view all of this information with different colors. Also, in the comparison table, we can see the molecules side by side. Or we can click through in one compound and then just go through the carousel to look at all the compounds, maybe flag some as favorites, and then just look at all of the data that is available. Now, all of these plots, they can be exported into PNG or PDF. Highlighted is the 2D scatter plot, but maybe I want to export this one. So you can select which plot you want to export, and you have the option to customize the axis as well, and then download. This will download high vector with quality graphic image. So it can be either PNG or PDF, and you are also able to export the data table and the data comparison table. Now, if you are using the visualization from CDD Vault, from within CDD Vault, you are able to save a session. Saving the session will allow you to come back exactly where you were before. If you are using the free version, then this option is not available. And of course, we can also share the session. So this copies the link into the URL. And then if you're writing an email, and you can simply paste that link. If the user is part of the vault and part of the project, they will be able to log in and see this view. Okay, so I just would like to mention that we also have an API. So the API allows users to connect CDD Vault with other tools or other clients. So these are just some examples, but there are other examples as well. And also the API allows the users to automate their workflows. So you can use all of these function calls to push and pull data in and out of CDD Vault in an automated way. Okay, so I think that's kind of what I had in mind to show you as an overview of the system. Now we have some time for questions. Yes, happy to answer questions, and thank you for your attention so far.
Thank you, Susana. So what Susana said, if you have any questions or any observations, impressions, it would be great to hear your thoughts. and Susana is here to answer your questions about the tools that are used by your colleagues from other universities and institutes, or you can maybe share if you're using something, if you see that this would be useful as well in the type of research you're doing at the moment. Please feel free. You can also still type the question in the chat if that's easier, instead of unmuting yourself.
Yes, whatever is easier.
Yeah.
I hope that you can hear me.
Yes, yeah, we can hear you. Hi.
First of all, I'd like to thanks to Susan and Katarina for this precious opportunity to have insight in your system, which is far more beyond than I expect. I know something about your systems since I saw you and we have opportunity to talk in Zagreb few years ago, but this is far more than I learned then, and thank you very much.
Absolutely. Thank you.
There are very many of informations, and I think that we need some time to digest them. I hope that this presentation will be sent to us, and that we will have opportunity to hear it again in slow time to have more insight in this.
Of
Very
course.
precious.
Yeah.
This is the very precious tool for all of us which are dealing with small organic compounds in the various fields of medicinal chemistry. Those who do all compounds that should be testing, and those who isolated them from the natural source, and also for microbiologists.
Absolutely.
Thank you very much, and it's great to see you here, Professor. Really.
Thank you. I just want to ask you,
Yeah.
are there any special equipment need to have this system, let's say, in our institute? What are the performances? Did this have a request for, let's say, station or server or something?
So, I mean, CDD Vault runs on the AWS. So all you need is internet connection in the modern and browser. So you don't need any equipment. So just a laptop or computer, and then either Chrome or Firefox or Edge, your browser of preference. As long as it's up to date, you'll get all the updates when you log in. And yeah, so CDD Vault runs on our servers in the AWS.
Thank you. I don't have no more question. And once again, thank you for this very good and informative presentation.
Thank you.
You're very welcome, and thank you all for joining. I just would like to mention that Susana shared how the visualization tool can be used, and I shared in the chat for those who joined a little bit later. Just in the chat, Zoom chat, you can see the link to the free visualization tool in CDD Vault. I just want to say, of course, yeah, it's limited in its use compared to the paid version. But once you upload your data here, you can see how scientists typically use it. Of course, it's a lot easier once you use the registration system in CDD Vault, where you register your entities and your assays. It's a lot easier to use the full version and possible to use the full version of the visualization tool, and it gives you a lot more. However, I think that this will be valuable to practice maybe on this free version. And this it doesn't only show you how to use it in academia, but also for the students. Once you start working in the industry, you know what to expect from modern tools in the industry. They're used in the industry. Like I mentioned at the beginning of the session, it's used by more than 700 organizations worldwide. So it's something that has existed for 20 years now, or a little bit longer. So it's not something new, but it's evolving with time as well. We're just keeping pace with it and keeping it modern and flexible, easy to use as well.
Absolutely. Yes.
We have, Susana, we have one question.
Oh.
Yes. So Miroslav asked if I understood well, each scientist can register and share scientific data.
Yes, yeah. So absolutely, yes. As long as they are part of the vault and part of the project, they will be able to register either in either a molecule or assay data, and then it will be available for all the other members of the vault. Sorry, I mean our what you mean in our public library?
Sorry, I'm not sure if I. Oh, he said, yeah, he's in a public library, so he cannot speak. But he just asked if each scientist can register and share scientific data. Just yeah.
Yeah, so
Yeah.
if they are part of the vault and part of the project, then the user can register either compounds or other entities and assay data in the vault, in the private vault, right? I guess that's what the question is.
Yeah, yes, I imagine just if each scientist, so once you have, maybe I can just mention that once you have your vault, we call it a vault, but it's actually your private database where you register your entities. Each scientist who works with you on the project can have access to that vault and actually register their data. And if you're, for example, an administrator, you can give them specific access, whether they can edit, view, or some other access to the vault. So yes, each scientist will be able to actually add data, register, and so on. It's a collaborative tool in the end, and it's all in real time, which means that once you register your data, everyone can work on it together in real time. That means the data is always up to date, that there are no different templates or different Excel sheets. If you're using Excel, for example, it's all standardized. So it helps you accelerate your research anyway. So yeah.
Exactly. And that just showed here. So you see that in this vault, in this drug discovery vault, in settings vault member, we can see all of the members in that vault. So all of these members are part of this database. But then in the projects, that's where we specify which members are part of what projects. So for example, in the CRO one here, we have these members, so they will only be able to see the data in that project. But then the drug discovery project has more members, so they will be able to see the data in the drug discovery project. And then there is also the access role. So some users can be read-only. Other users, maybe they can register assay data, and then other users, maybe they can also register compounds as well. Yeah, as well. And then the Vault administrator, it would be it's the super user in the account who can add the members, remove the members, and create the projects.
Maybe, I hope that answers the question. I don't know if you have any further follow-up questions, Miroslav, please feel free to type the question in the chat.
So the question is, if I understood well, we can order from other CDD Vault users their compounds. Yes, I mean, we can have a workflow where, so if everyone is part of the vault and part of the same project, then yes, we can have a workflow where you can order compounds. I guess when you say order is, yeah, I mean, I'm not sure if it's to synthesize, but you can assign a task or yes. And that's all customizable. Maybe I'll explain a bit better if I just show one of these molecules. Because that's basically all customizable. We can add a molecule field where we can have a note, maybe. That note can be a task. So all of these fields, they are customizable. So the Vault is customizable, and the Vault administrator can create these fields based on what information or what needs you have for the workflow. And that's something that we usually would discuss with you and would help you setting up all of these fields, because they are all customizable. What we see here can be different in your Vault. Okay. I hope that answers the question, but feel free to ask any other questions.
In the meantime, I just want to mention, of course, like I said, this is widely applicable in medicinal chemistry, but I know that there might be here those of you who don't work specifically in medicinal chemistry. It's also applicable in different areas of chemistry. So it would just need to be adjusted. So some fields might need to be set up differently to adjust to your workflow, but it's possible.
Mm-hmm.
Especially DLN would be probably valuable to capture all your research information or experimental information. What Susana shared, you can use it like Word, like type in free text or attach documents, images, tables, calculations as well. So, and it's all, what's most important, it's all interlinked. So everything is a couple of clicks away. You don't have to leave the system.
Mm-hmm.
And that's what most teams find valuable in these modern tools.
Exactly, yes. It's fully customizable.
Yeah. We'll then wait a couple of more minutes if any other questions come up.
Mm-hmm. Yes, no problem. Maybe I will just show in the meantime. This is just our updates. Our dev team continues to work. So actually, we do hear the feedback from all of our customers, and then we pass the feedback to the dev team, to the product management and dev team. And pretty much every month, sometimes twice a month, we have updates. And those updates, you don't need to do anything. You just, as long as you have the browser up to date, the updates will come up automatically in your Vault. But yeah, they are all captured in our web browser as well.
So also I would like to mention that what Professor Dan mentioned. I'm more than happy to send you the recording of this presentation so you can actually digest it and take a look again. And if any further questions come up, I also shared in the chat my email address, but I can share it again. I can just copy it and paste it again in the chat, just in case. You can contact me here either in Serbian or in English. And I will answer any of your questions if you want to learn more. Also, your feedback is more than welcome on this session as well. So I imagine that now we can maybe end the session, and of course, Susana showed you how you can track basically the history of your compounds from the start until you end your research project. You can see the whole history, and you can also use the graphs in your visualization tool to present maybe in your research papers and similar, maybe to show your successes or your outcomes in research. And like I said, feel free to ask any questions afterwards. So I want to thank you all for joining and for your time. I hope this was useful. Hopefully it gave you some insights on what your colleagues are using today. Thank you. Thank you all.
Thank you.
Thank you all.
Thank you, Elia.
Bye. Bye.
Thank you.
Bye.
Bye.